So I'm gone, obviously, again. So I'm going to be telling you about organismal reproduction, which isn't too bad because the ideas are really repetitive between each of the different types, but still. So we're going to look at mitosis, meiosis, and gene expression because we're going to come back to gene expression just because it's been a while since we've talked about DNA. So that's our goal. So first bit is the reminder, and that would be meiosis. So with meiosis, all we're aiming for is to produce gametes, which are haploid cells. So, just for the sake of saying it, haploid is a nuclear thing. It's not a cellular thing. So when we go through the process of meiosis, the only thing that we're really noticing between a haploid and a diploid is the nucleus is different. In some cases, the cell itself is going to be built differently, but the main thrust is you need to make sure that the nucleus is different. So the nucleus will have half the number of chromosomes it should have with the appropriate chromosomes being present. So when we want to have fertilization, different organisms will do this different ways. So if you go with the fungus, fungal lifestyles are actually rather fun in that, sorry, I'm shedding hair. You didn't need to hear that. So when you look at a fungus, for the most part, it spends its time in an asexual lifestyle, meaning it just produces spores. It goes through mitosis, and all it does is produce spores to make more of its kind. Only when the environment is bad does it actually enter into what we call sexual reproduction. So in sexual reproduction, what you're going to end up getting is the formation of two different nuclei. So you end up creating spores that turn out to not be similar. So we'll have our two different spores. What we'll end up doing is, so with these guys, it's a little bit odd in that they have two different fusion events. So what they'll do is they'll actually fuse their cells together. So it's called plasogamy. I can't talk right now. Maybe I should have been drinking water. So these two fuse. So the two cells will fuse. And what you'll end up having is what we call the heterokaryotic stage, which is there's two different nuclei inside of the cell. Eventually, the nuclei will fuse. That's karyogamy. So karyo is the nucleus. Gamy is marriage. So we'll actually fuse the nuclei. We'll actually have too many chromosomes. Then you go through meiosis, and now we're back to kind of where you were, which is we'll produce a whole bunch of different types of spores. So here, meiosis is used to produce the spores, and we actually shove all sorts of chromosomes together. Then we'll divide them up to create some variety. Fungi are odd, which is why it's worth pointing them out. If we look at plants, plants are a little bit more straightforward. Plants have two life cycle, or two parts to their life cycle, what we call the alternation of generations. We have the sporophyte and we have the gametophyte. The sporophyte is the plant that you think of. The gametophyte is the plant that forms gametes. And if I were to look at a flowering plant, the male and the female gametophytes are hidden inside of the flower. So we have the ovule and the ovary and the stamen, or in the pistil, excuse me. So all of that together creates what we would call the female gametophyte. Then you have the anther and the filament. That creates what we call the male gametophyte. And inside of each of them, there are going to be some cells. So here they're calling them sporocytes, that they're going to undergo meiosis. So inside of the ovary, what we'll end up producing is a whole bunch of different types of cells. And inside of the anther, which is where you end up making pollen, you're going to make a whole bunch of cells. Here, the pollen turns out to be a few things, but they are all turn out to be produced through meiosis. So pollen turns out to have gone through meiosis, and then they go through mitosis, and there's actually two different cells inside of a pollen Both of them turn out to be haploid. When we look inside of the ovary and we look at the cells that are found inside of there, they actually range from being either haploid, so we have antipodal cells and synergid cells, and we actually have the actual egg itself, which are all haploid. And we'll also have this weird thing called endosperm that turns out to be triploid. So when we go through and we want to fertilize a plant cell or to fertilize a plant, there's actually two different fertilization events because there are actually trying to be two different sperm that are produced. So when you finally get the pollen to land on top of the female reproductive part, so top is the stigma, the stalk is the style, you actually have two different sperm cells that are going to travel through a tube that forms. So the pollen tube that you can see over here labeled is just the connector. Pollen lands on the top, we'll have self-signaling. It says, hey, let me fertilize you. A giant cell will grow through from the top all the way down to the bottom of where the ovary is. So you can see massive old tube. The pollen tube, because it came from the why am I drawing blanks on these words? This is not good that I'm drawing blanks on words. When it comes from the pollen tube comes from pollen, everything inside the pollen tube is haploid. So the cell that ends up creating this pollen tube is haploid. Two nuclei get brought in, and what they are going to do is they're going to fertilize two areas. They're going to fertilize the egg itself, and it goes from being a haploid plus a haploid makes a diploid. And inside of that endosperm area, we have what we actually call polar nuclei. So there's two of them in there. You add the third sperm and you actually create what eventually becomes endosperm, which is actually triploid. It has three nuclei in it, or three sets of chromosomes, not three nuclei. It has three sets of chromosomes. And from here, we've actually created our brand new plant. This is only for angiosperms, so it's only for plants that produce fruit and produce flowers. That all this applies. All other types of plants, they have different reproductive styles. We're just doing this one because this is the one that College Board tends to like to ask questions about, and they said they probably are going to ask. If they're going to ask questions, it's going to be about angiosperms. So again, they have two sperm. The two sperm are going to fertilize two different parts of the plant. One is going to fertilize an egg. The other one is going to fertilize what we call the polar nuclei to create the endosperm. We'll get to that in a bit. So we have two sperm, two rounds of fertilization. That's why this is called double fertilization. If we look at us being animals, we actually have two different rounds of fertilization that we can go through. There's what we call internal and external fertilization. Both of them have their advantages, both of them have their disadvantages. If we have external fertilization, like this here is a sponge, you just shoot the sperm or shoot the egg into the water and you hope for the best. So what can we do? We don't actually need to undergo the process of trying to find a mate. We don't need to go and move. We could just stay where we are and just release sperm, release egg, hope for the best. So we don't have to worry about the mating issues. What we have to worry about, however, is will the sperm and egg run into each other? So we have no issues in terms of needing to expend energy to mate. We just need to make sure that mating actually happens. I need to jiggle my keys so the lights don't turn on. We have internal fertilization. We don't need to worry about if sperm meets egg, but we do have to make the effort to run into each other. So there's no neither good nor bad. It's just kind of, it is what it is. Animals don't have an alternation of generations, so the one organism will also be capable of producing the haploid cells, either the sperm or the egg, the gametes. So if we look at internal fertilization, because external fertilization, you just have sperm meeting egg, what we'll typically have, this is mammalian, but we'll have an area where we're producing eggs. So in us, we call them ovaries. And an egg will ovulate, so ovulation means release of an egg, and it'll be released into some type of tube. So in this tube, what hopefully will happen is sperm are going to meet it somewhere inside of this tube. I'll leave it to you to figure out how the sperm gets there, but we'll have egg and sperm, they'll run into each other. The goal is to only have one egg and one sperm meet. If you have one egg and two sperm, it tends to be lethal. You can't have two eggs with one sperm, so it's usually how the ratio of how many sperm into the egg. With animals, we seem to be intolerant of any mistakes. So it's one sperm, one egg. And from there, we'll start to fuse. The big question when it's internal fertilization is this thing, this egg that's now starting to develop and become an embryo, is this thing going to remain inside or is it going to be passed out? If it's going to remain inside of the organism, typically it needs to have some type of nutrient source, and that can vary because you could either make this egg very, very, very large, or you can make it so it's small and it actually needs a nutrient supply from its mother. So there are sharks that aren't like us, they don't have placentas, but they will keep their eggs inside of them. There are reptiles that will keep their eggs inside of them. So it's not like, oh, it's only us mammals who get to keep the living organism inside of us, the embryo inside of us. That's not necessarily true. It's just a matter of where are you going to have the best shot of keeping this thing, this brand new baby, alive. When we go for trying to reproduce, especially with mammals, we're complicated because we have internal fertilization and we keep the gestation period, so the development part, inside. So the result is we got a lot of stuff going on. And since this isn't physiology, I'm not going to go through every single itty-bitty detail. But what we'll say is we have lots of hormones inside of our bodies. Females, they turn out to oscillate, and that's because women have a lot more things going on than guys do. And what we get is a pattern between several hormones, so including estrogen and progesterone. FSH stands for follicle stimulating hormone, and LH, which stands for luteinizing hormone. These hormones are affecting different parts of the body, and that's what kind of makes this all really odd. So we happen to have these hormones, follicle stimulating hormone and luteinizing hormone. These are going to be affecting whether or not we are making an oocyte, so the egg, go through meiosis or not, and then if it's going to be released from the ovary, if it's going to undergo ovulation. The thing is, whenever you release the egg, whenever ovulation is, you kind of want everything to be ready for that to happen. So the result of that is, well, where is the egg eventually going to go? Well, it's going to land in a mammal inside of something that we call the endometrium. The endometrium, women, is the part that you shed during your period, during menses. So what we want to do is we want to have this egg, hopefully fertilized, land where it's supposed to land, which means the ovary and the uterus need to talk to each other, which makes it all the more entertaining. So ovulation, we have a correlation between luteinizing hormone and follicle stimulating hormone. This here causes the release of the egg. These also cause the production of estrogen, and also to a degree there's estrogen in there, or not, I said estrogen twice, of progesterone. Progesterone is produced by the ovary, which has to do with when ovulation occurs. Progesterone has nothing to do with the ovary. It has only to do with the endometrium, when the location where that fertilized egg is going to land. So we actually have hormones that are caused by one event that are used to perfectly time other events. So your menstrual cycle has to all do with when did ovulation occur and when is progesterone being produced or when is progesterone disappearing or what have you. There's a lot more that we could talk about with this, but here's the big deal. We have the ovaries, we have the uterus. They talk to each other so that they can have good timing. You want to have an egg being released. We want ovulation to occur when sperm will run into it, and that fertilized egg is going to land on the endometrium, land inside of the uterus, and not get dumped out. That's the goal. How do we do that? We use hormones. Once you have that fertilization, what we need to do is actually take this thing that starts out as one cell, which will then divide into two cells, which will divide into 4 to 8 to 16, 32, 64, 128, 256, 10, 12, things like that. Keep reproducing. What eventually starts to happen is we get different environments. So if I have one cell, the cells, everything's touching everything. 2 cells, it's all the same. The moment we start getting into like 16 cells, what we get is we get a layer on the outside and we have a layer on the inside. The outside cells are having different contact points than the cells on the inside. And one of the things with cells is what you touch matters, because that's where cell signaling occurs. What we get is an internal section where they do not know what the outside cells are experiencing. These cells here have different touching points than the cells on the outside. These cells right here on the outside, what we call the trophoblast, these cells here can touch the outside and touch these inner layers of cells. This other section of trophoblast is touching the outside layer, whatever's on the outside, and this big old cavity called the blastocele. So even then, these cells on the trophoblast have different environments. And if you have different environments, we're going to have different levels of cell signaling. Different types of cell signaling are going to cause different results, such as different genes are going to be turned on and turned off, because one of the things that can happen from cell signaling is we can get a genetic response. The cell could do something, but the nucleus might do something different as well. So why do we have gene expression? Because we have different environments. Different environments have different signals go through cell signaling differently. And the result will be each of these cells is going to start to become a little bit different. The fancy word for that is called differentiation. And it's all because these cells are in different environments. We can sit there and we can measure the fact that these things are occurring. So this is an example of different genes, and in different stages or in different environments, we could actually sit there and see what's happening. So these statistics here are saying, are these actually matching or are these not matching? So these are p-values, meaning the probability of them being the same. That's not that great, which means, yeah, these cells are being different. They are expressing their genes differently. And we can do this, and we could actually figure out, we haven't done it for everything, or anywhere close to everything, but for certain organisms, we're pretty good at this, where we can tell you what genes are being turned on and turned off at every stage of development. And it's because the organisms are small enough, they're simple enough, they reproduce fast enough, we can control them easily enough. They're what we call model organisms, where we can actually sit there and figure out lots of differential gene expression. When we look at development, gestation, across different sets of organisms, what we get are some patterns, especially if we're looking at similar organisms. So if we look at just animals, what we notice is there are developmental patterns that are very, very similar between all these. So these are a series of very famous pictures by a guy named Ernst Haeckel, this guy up here. For the sake of pointing out, there are some flaws with these. Namely, when you look at it, it's like, oh, look, they're all the exact same size. And the answer is, actually, no, they're not. They're not all the same size at all. So that's mildly misleading. Also, he exaggerated some of the features to make it look far more similar than they are. But the patterns are actually the same. Because if you look at all of these, eyeball in the same spot, we have this big old long tail, and we have these weird little slitty things right here. all of those turn out to be hallmarks of us being vertebrates. So having this tail-like thing, having these little slits along where your pharynx is, there actually turns out to be something that we call a notochord inside of there too. They're also the spinal cord turns out to be on the backside, the dorsal area, instead of being on the front, which is where it is if you look at arthropods. So we actually have these patterns that seem to show up. There's a phrase I gave you at the start of the school year that you don't remember, which is ontogeny recapitulates phylogeny, meaning how you develop tells the history of your evolution. Where did humans come from? Well, we came from things like reptiles, so we should develop like reptiles. Where did reptiles come from? Things like amphibians and fish. So reptiles should develop like things like amphibians and fish. So we should be developing similarly, not the same, but enough that you can sit there and say, oh, okay, I could see the patterns of fish and amphibians. So you'd respond with, well, what? That doesn't make any sense. Well, if we evolved from fish and amphibians in some way, shape, or form through some series of mechanisms, we should have similar genes, because the genes that these guys have are what were, meaning fish and amphibians, well, those are the genes that were available to produce the reptiles. And so whatever genes fish and amphibians have, that's all you had to work with in order to produce reptiles. And reptiles, whatever genes reptiles had, that's all that was available to produce birds and mammals. So there should be something similar, because we use the tools that the previous generations, meaning different species, before us had. We don't get to start from scratch every time. We have to use what we got. When we look at us being animals, if we look over the vast history of us, animals started out in the ocean, which means we were surrounded by salt water. If we look inside of our cells started in oceans. So the inside of our cells should be salt water. And what do we see? We have salt water. The inside of us is salt water. When we develop, fertilization, external fertilization is easier than internal fertilization. So it probably showed up first. So with external fertilization, where would it occur? It developed to occur inside of salt water. So when we got internal fertilization and creating eggs, which allows you to have better control over your future progeny, the environment that they need to be in would be what came before, which would be they need to be in salt water. So when you look at an egg and you were to test the watery environment inside of an egg, what you would notice is it's salt water. If we look at internal fertilization and having an egg inside of you, so being stuck inside of a placenta or attached to a placenta, what we get is salt water. We get these patterns. I think this is the last slide. Yeah, it is. So all of this stuff, all of this development is controlled by something. It's controlled by things called homeotic box genes. No one says homeotic box. We shorten it up to calling them HOX. So homeotic box. Homeotic meaning they seem to have the same parts, and when we looked at them, they're kind of chunked together, so we call them boxes. So we call them HOX genes. And what these things seem to do is they control how you develop. And if we look, there are patterns between all sorts of organisms, from C. elegans, so this thing here, Caenorhabditis elegans, here is a worm that we actually know every single cell of its body, and we know where every single cell in its body comes from. We look at Drosophila melanogaster, so this one here, this is the fruit fly. We know where all these body parts come from, and we can even start to match them up to the C. elegans. Brachiostoma floridi, so this is a lancelet. It's a tiny, itty-bitty, it falls under our category of organisms, which are called chordates, but they don't turn out to have internal bones, but they're called chordates. And they turn out to have similar patterns with these guys, with the worm and with the fly, which also turn out to have similar parts with a mouse and a human. We have the same genes, and it turns out that I could take a gene from a human and put it inside of C. elegans, and it works normally. I could take one from a mouse and switch it with one in a fly, so take one from the fly and put it into a mouse. The mouse develops normally. These are genes, these Hox genes, seem to be insanely uniform. And what we've learned is what these things do is they're not super special in terms of, ooh, they happen to be amazing genes, but they actually turn out to be things that control other genes. And we call those things transcription factors. And that's where we're going to be heading to next. We're going to deal with genetics and how we control genetics.